Artificial intelligence is revolutionizing the world in many ways, in many aspects of our lives. But in our field, this is one of the most exciting applications, particularly in this area of antimicrobial resistance when we are short of antibiotics. Artificial intelligence has come to the rescue to discover new antimicrobial molecules in the most unlikely places. That involves the environment and even our own human bodies. Today is actually the episode number 50, our AAC versions of Editor in Conversation. So we're very happy with this. And to celebrate that, we are going to talk about artificial intelligence and antibiotic discovery. And I am going to have an expert whose name is also César. So this is the podcast of the Césars today, celebrating our 50th episode. So with that, welcome to Editors in Conversation. The objectives of this podcast is to understand how artificial intelligence can help the discovery of new antimicrobial molecules and moieties, because not necessarily is a drug. Discuss antibiotic discovery and challenges to bring molecules to the clinic, and elaborate a little bit of the implications of artificial intelligence in this area of infectious diseases, particularly the discovery of antibiotics that we desperately needed. This episode is brought to you by the Antimicrobial Agents and Chemotherapy Journal that is available at asm.org/AAC. I'm your host, César Arias, Editor-in-Chief of AAC, and this podcast is supported by the American Society for Microbiology that publishes AAC. If you want to publish an AAC, and you do want to do that, it's better to become a member, since members get up to 50% off publishing fees. So it is really a service to the society and to humankind to publish here. Visit asm.org/membership to sign up. And don't forget to check our AAC issue available at the website with our outstanding papers on mechanisms of resistance, pharmacology of antimicrobial agents, and clinical epidemiology. We also have collections, including challenging clinical cases that have been very popular to learn how to use antibiotics in this era of desperate resistance. We recently had a very successful meeting called IMARI, was the first in its kind in Las Vegas, where the American Society for Microbiology and IDSC put together an amazing meeting. I'm telling you, the second version is going to happen also in Vegas at the end of January. So please put that in your calendar. The official announcement is forthcoming. So that's where we had the opportunity to hear our guest today, who is Dr. César de la Fuente, who is Presidential Associate Professor at the University of Pennsylvania and Director of the Machine Biology Group in the university. Welcome, César. Welcome to the show.
Thank you, and thank you so much for having me. It's great to have two Césars in a single show, and that's actually very rare, right? In academia, at least in North America, you don't find a lot of Césars, so it's really, it's really a pleasure to share this time with you.
Fantastic, and absolutely best way to celebrate our 50th episode. So in Imari, you give this phenomenal talk about AI and what you are doing to discover new antibiotics and opening new avenues of research. So let's start just because our audience is very diverse. So I think most people understand what artificial intelligence is. So why don't you explain to the audience how do you use artificial intelligence to discover drugs? And some of these drugs are, of course, antimicrobial agents.
Yeah, sure. Okay, so maybe let's start from the beginning. The way traditionally scientists, including microbiologists, have discovered new antimicrobial molecules is this very physical process where you go around nature and you go into these really fun expeditions. You dig into soil and you try to identify active molecules that might be present in that complex organic matter, like soil, right? As you can imagine, that's a process that is very painstaking, that depends on trial and error, and that it's unpredictable. At the end of the day, most likely, you're not going to find something that is, very efficient at killing bacteria. So instead of doing that, we decided, a number of years ago to to think a little bit differently about how we discovered new potential antimicrobial molecules. And we decided to conceptualize all of biology as an information source. So we decided to treat it as an information theory problem, where, you know, all the DNA of the world and all the proteins of the world, they're essentially a bunch of code, right? And DNA has 4 letters, the 4 nucleotides. Proteins have 20 letters, the 20 amino acids, and they're not that different from the alphabet that we use to communicate with each other. And so if we think of it like a soup of letters, then we can devise algorithms, including AI systems, that can mine, that can basically mine or explore all this soup of letters and identify letters that, when combined together, they make up words. And in our case, a word that has meaning represents a potential antimicrobial molecule. So we decided to basically think about the whole problem in a slightly different way. And so that's what we've done over the years. We've developed algorithms that can essentially mine the code of life for new functional entities, including antimicrobial compounds.
So, in order to decide to how to develop that code, I imagine, I guess in any computer we know it works, and then you do it. But just to design the algorithm, so how did you think about it, and how do you come about to design algorithms that were specifically for that particular function?
Yeah, that's a great question. So really early on, maybe around 9 years ago, we sort of had this realization that if AI were to be successful in complex fields like microbiology, right, is an incredibly complex field, we needed to come up with our own training sets, experimentally, using standardized conditions. So that's what essentially we've done. We've trained a lot of our AI models like Apex with standardized, high-quality experimental data. And in our case, what that looks like, it's a number of molecules. We typically focus on peptides and peptide design. So you have all these different compounds, and you do experiments to assess their antimicrobial activity against a number of target bacteria. So we have those different target bacteria and the different antimicrobial scores. So then models like deep learning models like Apex, what they do is they learn hidden patterns on the data, and they're capable of essentially, you give the model an amino acid sequence. From that amino acid sequence alone, it can predict antimicrobial function. So to answer your question briefly, we've developed our own training sets experimentally. A lot of microbiologists and chemists in my lab working really hard to then train the models. And I think this speaks to, I would say, the present of microbiology, not even the future, where it's this tight collaboration between some of these AI models and machine intelligence, and then the human microbiologists and human chemists and human biochemists in the laboratory. Is this really, I think, fascinating collaboration? And we need it, right? Because a lot of these machine systems, they have capabilities that are superhuman, right? We could not, we can't process that amount of data with our human brain. We can't identify those incredibly intricate patterns in the data with our human brain. We can do a lot of other incredible things with our human brain, but why not use some of these systems to augment our capabilities in the day-to-day in the lab?
This is true team science, Cesar, and I'm really amazed how I think 21st century science is exactly how you're mentioning, and the present lab, I guess, has to be a combination of experimentalists and computer biologists to get to the answers, the deep answer that you know. So one question that I had is why did you decide to go for antimicrobial peptides? I guess we know that antimicrobial peptides are the ancient molecules of the immune system against, the innate immune system is used. And that is something that is probably very attractive, but, it would have said probably something that will be difficult to implement since I guess the bugs are used to see that. So what are the reasons that you focus on particularly antimicrobial peptides?
I would say several reasons. One of them is that small chains of amino acids called peptides, or mini proteins, or essentially short chains of amino acids, are amongst the smallest functional molecules of life. So if you think about all the way you go from the beginning of life on Earth, some of those early molecules, they were polypeptide chains. And so from an evolutionary perspective and more, even origins of life perspective, I'm really fascinated by trying to understand how these chains of amino acids work, and how to develop methods and tools that allow us to program them in interesting ways, right? Not only learn how they work and how they evolved, but also, from a programming perspective, to be able to design peptides to do what we want them to do. So that's one reason. The second reason, like you very well mentioned, is that they're these very ancient innate immunity molecules. And so for a long time I've thought that there might be a lot more of these small peptides in biology that play roles that we had not anticipated, and that's just because we didn't look there and we didn't have the tools to look for such biological functions. And so that would be perhaps another reason. And the third one is that I think peptide design has an incredible future. For compounding on the prior two reasons, I think if we can learn how to program these small chains of amino acids, I think we can apply them not only as antimicrobials, but also to boost the immune system, or to treat tumor cells. And even beyond biotechnology, I think some peptides have semiconducting properties, or even as semiconductors, or for carbon capture, or for many other applications. I'm not even mentioning, of course, as GLP-1 agonists, right? Underlying a lot of the revolution in diabetes treatment and applications in obesity. And obviously insulin is also a peptide. And so I think I've always thought that they have peptide molecules have this sort of untapped potential, and if we can learn how to program them, they can play a huge role in the world to try to improve the world.
This is so cool, and this is such a great vision. So let's go into the story, because I mean you have really cool stories to tell, and fascinating publications into. So what don't you tell us that one of the first story, because that comes from a very narrow focus, and then to discover a much more broad application. So I love to hear it in your voice. So I'm sure the audience will be fascinated with that story. Please.
Yeah, sure. So a lot of this started, basically we were developing and applying very simple algorithms to try to find what we call sort of encrypted peptide molecules embedded within proteins. And we were applying these methods to identify, to sort of mine through one protein at a time. Let's say, a protein from the human body or something. With advances in compute power, we realized we could scale up this process. And so instead of mining one protein or one gene at a time, we realized we could do more in a systematic way. So we embarked on this journey to mine the human proteome for the very first time as a source of antimicrobial molecules in a systematic way. The human proteome is all the proteins encoded in our genome. So for the audience, you can think of this as essentially the genetic code that encodes who we are, right? And we had this rather simple scoring function. You can think of it like a pattern recognition algorithm, not that dissimilar to the ones that we use on our phones to recognize sounds or voices. And so the challenge here was that we had to mine through 42,000 proteins, because, probably a lot of people know in the audience, we have 20,000 genes as part of our genome. But in this case, we also took into account protein isoforms. And so in the end, the total number was 42,000 proteins that we had to sort through. We had no idea how long this would take. We actually thought that perhaps it would take a week, and we we had a number of guesses or bets running in the lab to try to guess how long this would take. And what was incredible is that everyone was wrong. It actually took 60 minutes. It took about an hour for the algorithm to essentially mine every protein encoded in our genome.
That is amazing, César. That
On
is amazing.
like 48 CPUs that we had at the time in lab. We didn't even have GPUs at the time, and so this really, I think it revealed to us in front of our eyes that it was possible to mine biology at scale to try to identify potential functional molecules, and I think it informed a lot of the subsequent work that we've done. And from a microbiology perspective, it enabled us to identify thousands of, we call them encrypted peptides, that the vast majority of which were not previously known to have antimicrobial properties or play a potential role in the immune system. And when we dug deeper into the data, the other fascinating thing, I think, is that over 98% of these thousands of encrypted fragments were derived from proteins that have nothing to do with the immune system. Their functions in the daily job has nothing to do with host defense. And so that observation, it led us to propose the encrypted immunity hypothesis. So we think that there is a lot of encrypted layers of immunity or hidden layers of immunity in the form of functional molecules, which can be small peptides, for example, that get released only upon need, for example, in the context of an infection or in the context perhaps of physiological stress. And, so this encrypted immunity hypothesis is still a working model. We still need to do a lot of work to kind of stress test this hypothesis, but I think it's a really interesting one, because if you think about it, it's sort of unbelievable that we don't die every 5 minutes, right? We're exposed to millions of microbes every time we leave our house, or even in our house, or I'm here in my office right now. There are so many microbes around me, but yet we don't get sick all the time, right? And so to me, it's not entirely, that surprising that perhaps our immune system has all these tricks, right, to keep us healthy.
So basically, you kind of unveil a new immune system. This is a massive thing, right?
That's the hypothesis. That's the hypothesis.
Right? That's absolutely fascinating. And to me, particularly as a clinician, and where I am on service right now and seeing patients in dire states with bad infections, you see that there is some sort of sweet spots when all of that fails, either because we do something for humans, for the patients, or the organisms are capable to overcome certain things. And I want to think about that concept in that you just mentioned about our symbiosis with the microorganisms. And based on that hypothesis, if it turns out that we have a sort of a hidden immune system, in some ways is also evolutionary trained based on what we live with. Isn't that something that is possible and can vary according to where you live or what you eat or where you are used to?
Yeah, potentially. We think one way by which these encrypted peptides get activated or released from proteins is through proteolytic cleavage. So you have chewing enzymes that essentially chew the protein and liberate or release these encrypted fragments. But again, where do those proteases come from? They could come from the host, from our own body, right? We produce proteases. Or they could come from microbes, right? Right? The microbe could produce a protease that then cleaves a human protein, releasing a fragment that then has antimicrobial properties. So this is an experiment that I've been wanting to do for a long time. Maybe somebody in the audience wants to do this project. I think it would be a fascinating project where you can test proteases produced by members of the microbiome, right, that have co-evolved with us, that then cleave human proteins to release fragments that perhaps are active against pathogens but not commensals. And so it could be a way of microbiome members sort of helping humans to fight off other microbes. So it could be sort of this cross-kingdom warfare where microbes, I mean, this is all a hypothesis. We have no proof of this, but I just think it would be a fascinating science project, a research project where there might be microbes as part of our commensal communities that might be helping the host humans against other microbes. And if you think about it, that's if that were to be true, that would be kind of incredible, right? That a microbe would help a human to fight off another microbe. But anyway, I'm just sort of daydreaming here. This is just an idea that I have for a project, but we haven't done this yet.
It is fascinating. And imagine that in the context of you giving antibiotics to a patient that goes into the immune system that disrupts many of these relationships. What relationships will remain? Would that beneficial? Who will benefit eventually of this?
Right.
So moving on, you actually move out from the humans and you just went to nature and even prehistoric information. Tell us about that. And you start discovering antibiotics there. I mean, this is fascinating.
Yeah, sure. So we did the human genome, human proteome work. We found all these new sort of peptide molecules there. And so this really sparked a lot of new ideas and hypotheses in my lab, one of which was perhaps these are conserved. They're perhaps not just confined to the human body. Perhaps they're conserved throughout evolution and across the tree of life. And so that took us to see if we could find some of these encrypted fragments or encrypted molecules in our closest ancestors, which are Neanderthals and Denisovans, essentially archaic hominids. So that was the initial motivation. But the second layer of motivation or rationale for this was that I started reading a lot about this. And as it turns out, the vast majority of life that has existed on our planet is now extinct. The vast majority. So whatever we see around us today, the life forms, including humans, but every other life form that we see in nature is just a really tiny fraction of everything that has ever existed. But the vast majority of what has existed has disappeared through different extinction events. And so how can we pretend to know anything about evolution or biology or microbiology if we don't know anything about that, right? And so that was sort of like more a motivation from a fundamental science perspective. So that was the impetus for looking at the genetic code of Neanderthals and Denisovans, who are essentially our closest relatives of Homo sapiens sapiens, to see if we could find some of these encrypted molecules there, right? And in the process of thinking about how to even begin how to do this, we, through a number of brainstorming sessions, we, along with my team, we came up with this conceptual framework that we call molecular de-extinction, whose ultimate goal is to identify functional biomolecules. This can be small peptides or small proteins or metabolites throughout evolution, right? Throughout the whole sort of tapestry of evolutionary history, to then make them in the lab through chemistry or recombinant expression, to learn how changes in those functional molecules over time influence their function. And that function can be an ability to kill bacteria or target tumor cells or boost immunity or, operate, interfere with metabolism or bind to or operate as a GLP-1 agonist, right? It could be anything, really. So we applied that framework for in this particular project. We developed a machine learning model that could predict essentially encrypted fragment release from Neanderthal proteomes, for example. And then that enabled us to identify potential antibiotic candidates encoded in the genetic material of these ancient hominids. And then we made some of those molecules through chemistry, and we can actually make them in the lab. We have these robots that do solid phase chemical synthesis, so we can sort of recreate these molecules, some of which, by the way, As far as we know, they're not present in the world today. So some of them, when we do sequence homology and alignment, we don't see them in any proteins in contemporary biology. And we saw that some of them were effective at killing bacteria in vitro. Some of them were effective at reducing infections in mouse models of infection. And so that was fascinating because it was not only a computational curiosity. We actually, some of these were actually functional in the lab, right? And so anyway, from that project, we learned we could use AI to mine ancient genetic material to identify functional molecules. We learned that we could then make them in the lab, and we learned that they were active. And so this gave us it was the proof of principle of a lot of the work that we did subsequently, essentially.
Yeah, and I mean, this is absolutely fascinating. And we sort of understand extinctions in a macro wave, but just at the protein level, what you're saying is absolutely fascinating, that we lose certain templates and certain functions with these events. And you went even to the dinosaurs, didn't you? You started looking at genomes of. So tell us about that and what you found.
Yeah, so we did the ancient hominid work, and then that we were able to surface functional molecules. So then we said, okay, well, let's try to scale this up one more time. Instead of just confining ourselves to archaic hominids, why don't we sample through all of ancestral biology, right? We went to the drawing board. As it turns out, ancestral biology, we actually have more information than I thought. So what ended up happening is that we had to develop a new AI model. So that's when we developed Apex, which we were talking about earlier. And Apex was pretty incredible because it enabled us to essentially sample through all of ancient biology at scale to identify antimicrobial peptides. It took us through a journey through the Holocene and the Pleistocene. So yeah, it enabled us to look into the genetic material, let's say, of ancient penguins that were extinct in the 50s, so pretty recently. Magnolia trees, beautiful, like huge magnolia trees that disappeared throughout evolution. Ancient elephants, woolly mammoths, giant sloths, and many, many other creatures, all the way, like I said, from the Holocene and the Pleistocene. And a lot of these, it really gave us really highly potent antimicrobial compounds, right, when we tested them in the lab. And we think of molecules essentially in my lab as these sort of documents of evolutionary history, where we can extract potential new medicines or potential new therapeutics, but also more from a fundamental or foundational science perspective. We're interested in learning how evolution did things. Like, why did evolution make that particular mutation here? Why did that happen throughout the course of evolution? And what did that mean for the function of that molecule? And so now we have a number of projects in the lab where we're tracking the natural history of some of these molecules all the way to the present day. And so we're really fascinated also by that, the perspective of trying to understand a little bit better how evolution actually worked, right? The evolution is this, we have access to this, the largest optimization experiment, right, that we know of is evolution. So why not try to learn from it?
Yeah, that's absolutely fascinating. So I want you to comment about the computer power when you started this journey up to now. I mean, how has that evolved, and with everything that we see now in AI in the news and the power of the chips and all that? So how do you manage to acquire that expertise and increase it and scale it, as you mentioned, and create new algorithms? And how do you see that going into the future?
Well, this has been a whole revolution, but in and of itself, right? We started with a couple of dozen CPUs. We did the early human genome, human proteome work. We published that in 2021. And all the work that we've done after that has relied on GPUs, right? And so I think the advent of GPUs has enabled us to do, a lot more projects and develop AI models that are a lot more compute intensive, right? Deep learning models, and now we're also developing foundation models for a lot of the work that we're doing. And to train a foundation model, you need a lot of GPUs. That would have been impossible to do with what we had even 5-6 years ago. So one thing we've had to do is we've had to fundraise and acquire funding to be able to build a GPU cluster, which is what we really rely on that for 90% of our work right now. And that is not cheap, right? Because these GPUs these chips, right, are one of the most coveted things in the world right now. And so, first of all, it's really hard to get them because the inventory is very low, and then they're quite expensive. You can pay for one over $100,000 for a single GPU node, and so. But we've been able to build our own GPU cluster. We have direct access to it, and so we have certainly taken advantage of this revolution and this transition from CPU compute to GPU compute.
That's fascinating. And so you're in the field. What other people are doing with AI to discover and antibiotics? What are the other approaches that you can tell that sort of excite you as well?
I mean, there are people applying AI systems more for small molecule discovery work. Jim Collins over at MIT is doing work, for example, along those lines. There are people in companies doing interesting work. Genentech recently published a paper also more oriented towards small molecules. So, I wish there were more people working on antibiotic discovery, to be honest. I think we're losing talent. There was a Nature piece, I think, last year doing an analysis on where the field is going, and over time we're losing young talent because there's less funding to do this sort of work, even in academia. And as you probably know, big pharma has mostly stepped away from developing antibiotics because it's not a profitable business. And so I wish there were more people doing this kind of work. And it worries me, right? Because if you look at the grand scheme of things, today, in 2026, bacterial infections, superbacteria that are multi-drug resistant, are associated with about 5 million deaths per year around the globe. By 2050, that number is projected to double or close to double, becoming the number one cause of death in our society. So I think it's the greatest existential threat to humanity is coming up with solutions against infections. And yet, almost nobody's working on this, right? So to me, that's this sort of incredible paradox, right? So maybe people in the audience, we certainly could use a lot more young minds and driven minds that are maybe in high school right now or starting college, to kind of join these efforts. And the other thing I'll say is that microbiology, antibiotic discovery, people working on AMR, antimicrobial resistance, these are becoming incredibly transdisciplinary fields. I remember when I was doing my PhD, if you did microbiology, you focus on one particular pathogen, and that was 99.9% of people, that's what they would do, which I think is fantastic. And now I feel like the field has a lot of these fields have welcomed people from computer science, from chemistry, from engineering, even from physics, come along, help us move these concepts forward. And so in the audience, there are probably a lot of microbiologists and microbe enthusiasts. But in addition to that, if there are people from other fields, I would say, come on, join us in some of this in this journey to try to provide solutions and try to tackle, I think, one of the greatest problems that we're facing as a society. So if you have people that care about the world and about humanity and about our future, I can't think of anything more beautiful and more fulfilling and more exciting to be and more challenging to be working on right now.
Absolutely. And you're probably going to receive 100 applications for postdocs after the podcast.
Sure.
So, so as a physician scientist, I want to ask you what you think. I'm on on the other side where I see people in front of me with I don't know how to treat.
Yeah.
And I see this beautiful science being done on the other side, but it is really hard for a journey for anything to get into a patient, and that's something that I'm very passionate about, because at the end of the day, my motivation really throughout my career has been the patient and what I see every day.
Mm-hmm.
And you're going back to the lab and try to do something about it. How do you envisage that these discoveries can be better sort of funneled into a clinical pipeline that reaches the people who actually need this sooner rather than later?
That's a very challenging. It's a very challenging problem, right? And the reason for that is that it's really hard to attract investment from VCs, for example, to do antibiotic development. That's because the market is broken, right? There is no return of investment at the end of the day, right? Even for successful antibiotic companies. So the incentives for development are just not there, at least not through company formation or startups. So that's one thing. What we try to do in my lab in academia is try to think about the whole problem a little bit differently, or out of the box, or in an unconventional way, perhaps. And what we have done is we've flooded sort of the preclinical pipeline now. Typically, it can take years to discover new antimicrobial molecules with some of these AI models and that are trained on experimental data that is high quality. We can discover now, by mining biology digitally, we can discover things in minutes right now, right? Potential candidates. But of course, and so instead of years, now we can do that sort of thing in minutes or hours, which I think is already really interesting. But the challenge remains of how do we take these preclinical or interesting candidates, how do we modify them, or how do we take them through IND enabling studies. So IND stands for investigational new drug studies, which is what you need to do successfully prior to going into phase 1 clinical trials. And who's going to fund that, right? Because if there's no investment, it's really hard to fund that. So it's this sort of incredibly challenging thing. That you're really going against the current throughout the whole process, right? Of trying to develop an antimicrobial asset or an antimicrobial lead candidate. So I don't have a good answer. We've spun out a company to try to take into the real world some of the discoveries that we've been able to make, but we know it's going to be incredibly challenging. So let's see what the future holds.
Yeah. So my last question is thinking about the future and about this fascinating things, like with the advent of quantum computing and all that. So what would be your next level of, you say now it takes me 8 minutes, now it takes me 2 minutes to discover an antibiotic. So do you see that going in that direction?
We actually have a collaboration applying concepts from quantum computing to some of the stuff. Is really early stage work. So I don't know where it's going to take us yet, but we're beginning to delve into quantum a little bit. I think the potential there is huge. It's just very hard because we don't fully grasp how the systems function, and so that is sort of probably the earliest stage research project that we have right now in the lab in collaboration. So perhaps in the next year or two we'll have more updates to discuss there. But I think I'm always excited to try new technologies, and I always think that the best way to innovate is to try to enter the intersection between fields. I think at that interface is where you can unlock some of the big discoveries, or at least sort of unanticipated discoveries. And I'll say, I think this is the best time in history to do science. We have all these incredible tools. We have, I'm not only talking about machine learning and AI systems, but high throughput screening methods. We have, we can sequence genomes at scale. We can read them. We can write them. We can do engineering of microbes. So I really do believe this is the best time in history to do research, and I hope a lot of people in the audience also think about research as a vocation, as a way of life, and to try to move the world, to try to improve the world a little bit. I think that at least that's my goal, and the goal of a lot of my team members, that, by the way, come from all around the world. We have people from many different countries, including the US, of course, and we have people we have computer scientists, we have engineers, physicists, chemists, microbiologists, biologists, all working together, and they all think differently about the different projects that we're trying to tackle. And I love that. There's this magic that happens when different people think differently, and then they try to provide ideas to move a research project forward. And I really have actually proof of that. So when we have people applying to come to the lab, I think the more creative papers that we've been able to publish, you look at the author list, and I can literally say, this is a computer scientist, this is a microbiologist, this is a chemist, this is a biologist. And that is, I think, proof of that. I think magic and the creative magic happens when you put together people that are thinking from different lenses and different perspectives about a project or about a problem.
Beautiful. With that, I want to thank you, César, for this beautiful conversation. Super inspirational, and I'm sure a lot of people are going to be very excited about this field and continue to work on this. This is, we will continue discussing this in IMARI, our prime now national meeting of antimicrobial resistance that is going to take place in Vegas in 2027. Thank you, César. Thank you, everybody, for listening to us. This is César Arias, Editor-in-Chief of AAC, signing off.